time to get started with this seminar. My name is Jesper Wengel, and I have the pleasure of introducing Kira Astakhova, who is an associate professor at the Department of Physics, Chemistry and Pharmacy. And she has been associate professor since 2012. And Kira is a prime example that we here at SDU have a lot of younger researchers with an incredible talent and potential to really become an internationally leading researcher. Kira got her education from Moscow, from Russian Academy of Sciences. In 2004, when Kira was 19 years old, she co-authored her first research paper, and already 1 year later the second one. And today she is author of 35 research papers. We have been so fortunate to have Kira as a collaborator here since 2006 as a PhD student. She came here for some time, then she went back to Moscow, finished her PhD, and then as a postdoc she came back to the Nucleic Acid Center. She has been developing her own research, and she has established, for example, collaborations with industry. She has established collaboration with research groups at the University of California and at Stanford, where she is also regularly a visitor. So Kira is really doing very well. She has she's obtained a Server grant, and now she is Willum Foundation, a young investigator, with a 10 million krona grant to develop her research. And that is in between organic chemistry, modeling, biotechnology. She is a true chemical biologist, and what she will tell us about today is diagnostics, so detection of nucleic acids in very specific way. So, Kira, we look forward to your lecture.
Thank you, Jesper. I hope to live up to that presentation. Yes, my name is Kira, and I've been here for the last 11 years, working with nucleic acids with a primary focus on diagnostics, which I want to present today. And yeah, so to outline what I want to talk to you about today is, I want to start by introduction and discuss why we need to know DNA and RNA sequences, including mutations, for biological species. How is it done now, and what can be improved? Then I want to present you our pilot study where we develop a new, I don't want to call it sequencing method, but it is actually a bit of different sequencing approach than the one that's been used now. And this is done by click chemistry, which I also want to talk about. And then I will finish by perspective and a bit of discussion, what, in a bit broader perspective, what this all is about, and discovery, ultra-fast specific discovery of nucleic acid sequences, what it means for the society and for the research community. So you are welcome to stop me at any moment if you have questions. So yeah, I want to start, like, when I say us, what are us? I have a research group here at the University of Southern Denmark, and we do interdisciplinary research focusing on nucleic acids, their analogs and interactions. We collaborate a lot, so you also in Denmark and across the globe. I'm very happy about collaborators in Denmark, and you already met Jesper, right? There are some other guys which I'm really happy to collaborate with. And yeah, this is just for this project, those who have been contributing. And we also work with industry, yes, that's true. This project in particular is done in collaboration with Thermo Fisher Scientific and Promega. Very nice collaborations we are very proud to have. Yeah, so this is all pretty much my background. Jesper already told about. So I'm Russian. I come from here. Yes. When I was quite young and was interested about nucleic acid chemistry, I got to Denmark, and that was the time when Nucleic Acid Center has been established, and that all influenced my research a lot and inspired actually this project too. Under my undergraduate studies, I worked for Oxford GenTech, and as a synthetic chemist already got this inspiration from all the sequencing, developing stories, genotyping, and stuff like that. And then I was also quite curious what happens here in this part. So I worked here in Boston and then also in California in different places. So, and I still work actually at all of these places once in a while, and I get a bit different experiences from here there. And my key interest, yeah. So this was, I just wanted to show you that this is the place where I graduated from. It's built as double-stranded DNA. So I actually always thought that was my office here in this third, yeah, it's on the third floor. So it was kind of, I don't know, meant to be, yeah, but it's just funny building as a double-stranded DNA. Yeah. And my research interest, as Jesper already said, is chemistry primary. So I have a training as a synthetic organic chemist. But then I shifted a bit to biotechnology and biomedicine. And for the last many years worked with nucleic acids, integrating these disciplines. So I was so lucky to study in 2000s. 2000s is the boom in nucleic acid research and biotechnology. I call them NA, I hope it's okay, nucleic acids, yeah. So it's all this time when in the beginning when I just got my master's, no one really used PCR for much things, right? And then all this happened. Amplification, sequencing, the Human Genome Project was finished in 2003. And then late 2000s, imaging including super resolution techniques. All of this happened very fast, and I remember like very getting excited every year from new techniques coming, new discoveries. And yeah, so it all contributed to my research interests. And I did organic and medicinal chemistry and then shifted to bioorganic chemistry for fluorescent nucleic acid probes that we did in Oxford Gene Technology. So already at that time we applied fluorescence for sequencing of nucleic acids. And then later on I worked at the hospital in Moscow where we clinically applied sequencing and PCR microarrays also for immunology studies. In 2009 I started doing postdoc here at SDU, integrating all of that a bit together. And from 12 to 15 I also worked in States, where my main focus was actually autoimmunity and oncology, so applications of all of these technologies in medicine. So it's quite a mix, one can say, but yeah, I'm going back now. So just to kind of get an idea, where are we now? So how many of you have heard about the genetic code? If I should introduce, yes, right. Genetic code, yes. So quite some, right? But so this is the first part, which is very important to what I'm going to talk about, is that A pairs to T and C pairs to G. These are nucleotides and they form the genetic code. And there is another nucleotide, an RNA, it's called U. And this is essential of living species, of inheritance, and I guess you also saw this part, that DNA gets transcribed to RNA and then it translates to protein, and a bit recent information saying that not all RNA becomes protein, but also some RNA molecules are regulatory RNA, which regulate other RNA and never get to protein. So I hope all of you know, or most of you know this part. A big part of my work is the discovery of the detection of mutations. So sometimes things happen in genetic code, and there come alterations. So the standard, this ATCG correct sequence, it gets some variations. And an example could be A becoming, instead of here should be C, but it becomes A, and then it doesn't base pair. Or for example, TT. We call it a mismatch or a mutation. And these mutations, they happen all the time while in living species. Many of them, they're called neutral polymorphisms. They do not not have biological effect. But some of them, especially in oncology, for example, they have a crucial impact if they occur in oncogenes, for example. And then the patient can develop cancer. So some of them are important to detect, some of these mutations. Yes, nucleic acid chemistry, biophysics, and biotechnology are my major fields of knowledge. Here you can see the sequence of nucleic acids. So it has three main parts. It has the nitrogen bases, this ATCG genetic code. Then it has also phosphate backbone and sugar. And because I am a chemist, so when I look at this, I see a chemical. I see it's very advanced, it has many different groups, but this is a chemical. And it has quite different, chemically, these are quite different parts. The first one is nucleobase. It is a heterocycle, so the type of organic reactions that one can do on base is quite specific. When we move to sugar chemistry, this is a whole different story. A part of my group is working now on sugar chemistry. They're not really happy about it. It's quite a tricky one. And there's a whole different reactions what you can do on this part of DNA and RNA. And the phosphate chemistry is also quite different. It can be done enzymatically, it can be done chemically, you can combine both of them. And the phosphorus chemistry is the one which cells are running all the time to polymerize this DNA and RNA and to create new cells with new genomes. So for me as a chemist working with nucleic acids, there's been always a lot of different methods, reactions we could do here, and never ever I got bored in the lab working with them. These are really great molecules, I think, DNA and RNA. And in cells, they form when the two of them meet and there is a base pairing, they form duplex. And on the slide, you can see this is ideal bDNA, B-type DNA. And this is the structure that has been discovered in 1953 by Watson and Crick, explaining the recognition between nucleic acid polymers. And as I already said, A pairs to T and C to G, and this is why this complex is formed. If there are alteration, variants, or mutation, or whatever you call it, then there is no base pairing here, and the duplex structure slightly changes. What we want to do in our work, we want to detect this change very specifically, and with a high sensitivity as well. Here I want to say, so this key paradigm of work of nucleic acids within the cells, and why I think, I believe it's so important to know this sequence very precisely. It's because what we call primary structure of nucleic acids, so just sequence ATCG for DNA, it encodes the secondary structure, so this duplex formation, for example. The way it looks, the way the geometry is formed, depends completely on the sequence. And this secondary structure is a foundation for tertiary structure, which is could be quite complex. For example, for some RNA, quite complex, three-dimensional structure. So once again, so primary structure is the fundament for function, actually, for formation of functional nucleic acid complexes and their interactions with proteins, with peptides, with small molecules. It is very, very important to know this sequence. And having done some chemistry or having modified this sequence, you actually influence this part and then that way influence this part. Keeping in mind what is the function of nucleic acids in cell, this is a big power. If you can affect the inheritance material of the cell, this is very intriguing aspect. Yeah, so, and the same concept works also for proteins, so also the primary structure encodes the secondary and tertiary. And this is also the reason why there was so much attention on sequencing, on decoding the primary sequence of nucleic acids. That was done in order to understand their secondary and tertiary structure and understand how it connects to function of the particular sequences in cells. So sequence is storing brief. I don't want to give you lots of information about it, but from very beginning of the nucleic acid technology, there were attempts to sequence them. And the first methods were degradation methods, so it just purified the nucleic acid from the biological sample and then degraded it, cut in small pieces by other chemical or enzymatic method, and then tried to analyze what the pieces are, and then put it all together. But quite fast the community found out this is not very efficient, you need a lot of material, it's very biased. So it shifted to the synthesis methods, or sequencing by synthesis, which is mimicking normal synthesis of nucleic acid polymer itself. And having a template which is not degraded polymer, then step by step creating a complementary one, using the same genetic code, A pairing to T and C pairing to G, and identifying every single base which is included, allows to decode the sequence. And this is what modern sequencing is doing. It has a lots of name, again, as mutation, has lots of names. It's called Illumina, next generation sequencing, sequencing by fluorescent terminators, and so on and so on. But it is step by step incorporating complementary nucleotides to the template. And at each step, it detects it by fluorescence, by color. For example, C nucleotide, C letter will give this one, G another one, A third one, and T fourth one. And then the machine records color at each particular step. And that's how a final sequence is given. If there is a mismatch or mutation at some part, the sequencing will tell about that by incorporating this particular letter. Yeah, and the read, this is the key term here. The read is the length, simply the sequence of the target DNA that has been sequenced. This is called read. And this is all done now alphabetically. I don't think that anyone here, maybe someone remembers sequencing gels. I've done sequencing gels myself. I don't think anyone who studied late 2000s have ever seen them, this huge, very large gels. So it's all done now alphabetically. And even, for example, Illumina is the platform for sequencing. What is important here also to say is that it's all done enzymatically. So it's an enzyme, again, it's all mimicking cellular processes. So step-by-step enzyme incorporates this complementary nucleotides. There is an issue with nucleic acids that typically in the biological samples they are present at very small amounts. And how to deal with that? So this is an important biotechnological method that is being used now. It's called amplification. And this is the, for example, polymerase chain reaction, or PCR. It's not the only one, but this is the one which is used most often now. It's the biochemical technology to amplify signal a few copies of DNA, generating more of it, basically. So, for example, let's now think about the sample. This is some bacteria or virus that has a nucleic acid in that we want to sequence. The first thing to do is to purify its nucleic acid material. And then, because it's such a small amount, usually, we need to run amplification, also using enzymes. And then we have more of this sample, which is subjected to, for example, Illumina sequencing, and that generates this data. So looking into this pathway, first what I see is it's multi-step process. It has at least 1, 2, 3, right? 3, but actually more than that, 3 steps. But the problem with this method is that when we have the sequencing data, actually this is not sample A. This is the data for station D and this path. So the workflow already has affected the initial sample quite a lot in some cases. And having this data in hand, we cannot say that we know, I mean we can, but there is definitely a risk that this data is biased, simply because of very long workflow. And this is the PCR just to show you how that works. This is initial sequence, then it gets denatured by heat simply. And there's this polymerase which can work under high temperature that creates more and more of this DNA. While it's creating more and more DNA, it can make mistakes. And it actually does make mistakes sometimes. And especially for detection of mutations, when we want to be very accurate, this can be a big problem. In this project, which I want to present to you today, we are talking about sequencing of RNA, actually, not the DNA, but RNA. So RNA can be also sequenced by Illumina, by next generation sequencing, but in order to do that, first it has to be reverse transcribed to DNA, and then sequenced. That adds one extra step, which applies enzymes. And here's just a sum up. I don't want to go through all these steps, but this is a workflow for RNA sequencing, which is right now recommended by Illumina technology. It's a lot of steps, and they mention on their own description of this process that be aware of high risk of misdiagnosed mutations. And our question was, again, to sum up, what's the problem with next generation sequencing? This technique is there, and it's great. It made very big impact on the many communities, right? Also in clinical work. So please don't get me wrong. I'm not going to criticize it and say that it all is so biased, makes mistakes. It is there and it's doing a great job. But the problem with it is that not a single step of it can be done without applying polymerases. So it's all fully dependent on work of enzymes. And these polymerases, they make mistakes. And this is proof now for sure that there's approximately 3 to sometimes even 7% of incorporated nucleotides while you sequence can be mistaken. So not all DNA and RNA sequences can be amplified simply. So I already said that in order to get more material, we need to amplify. But not all sequences can be amplified. Some of oncogenes, human cancer-related genes, they cannot be ever amplified by enzymes that we have now in hand. So how to detect them? It's very long and expensive. I wrote here expensive. The cost goes down every year, but still it remains being quite expensive workflow if to, let's say, use it in daily work in clinics. Especially for RNA sequencing, results are sometimes highly reproducible. So if we run the same sample a couple of times, the sequence that we obtain is quite different. But my main issue with this method to detect mutations and in general to sequence nucleic acids, that it affects initial sample. So again, if you look here, so sample A, then you get B, then you get C, then you get Z, sometimes E, sometimes F, and then you get the sequencing data. Who says that this corresponds to what I'm interested in, which is A? So this is a concept which one also calls direct methods. Direct method works here. It detects the sequence there where it originates from, without doing too much of manipulation. We are not the only one who realizes this problem with the ongoing sequencing methods. There are some existing alternatives already. And one of them I want to present is called nanopore sequencing, the technology which is now being developed in Oxford University. It has a totally different principle from enzymatic sequencing. It uses nanopore. Nanopore is a protein-based pore. And the DNA, you can see here, it gets through it. And while this DNA gets through the nanopore, it creates the current change, which is specific for each particular ATCG. And that's how the sequence is detected. It's very inaccurate. And even the producer now says that it's only 80% accurate. So in case of our goal, which is detection of mutations, it's not working right now. There are constantly new updates, new papers on that, which claim to improve the specificity, and we hope that it's gonna at some point do better. But right now, this is what it is doing. And the another interesting alternative, which also we are using, is the sequencing by hybridization probes. So if you imagine now, this is DNA target, and we can create a probe which binds to it, to a specific region. And then this is an example which is called branched DNA method. So there is a signal amplifier which has an extra signal to detect this event. Then this is a method to sequence as well. And these are two alternatives which exist right now. And I want to start now the discussion of our own work from here, so from hybridization probes. Our goal is to make the direct methods for detection of nucleic acids. That means no amplification, no reverse transcription. Just get directly into the sample, add the probe, signal amplifier, and get a yes or no right away. And why is it good to do that? It's very robust technique. It's very simple workflow. It's fast, it's cheap, and it allows what is very important for us to detect SNPs specifically. Yeah. And we have some preliminary data which have been done for quite some time ago that when we modify chemically nucleic acids, then we can actually create such type of probes that bind very specifically and that can also discriminate mismatches or mutations. By applying fluorescence microscopy, you can do it very specifically and also with a high sensitivity. Right now, one can do signal molecule resolution actually. But this is not needed usually. Usually, DNA and RNA are present at some copies per sample, so it's not, there is no need in super resolution detection. And chemistry of nucleic acid analogs has been developed here in Odense very well, and we use a method which is called oligonucleotide synthesis, or here it's DNA synthesis. We can create reagents. It's from the paper that has been published by this group here, working in Odense, where we can incorporate modified nucleotides into the DNA. So if you see here, this is actually synthetic DNA probe that can be created by this method. There's another method also I have to mention. It's called bioconjugation. Bioconjugation of oligonucleotides, for example, by click chemistry. This is an organic reaction, but it is very good for DNA and RNA chemistry because it can be done in water. Some very good chemical methods, they do not work in water, and they're not applicable for nucleic acid chemistry. So this click chemistry allows to take one piece of DNA, the second piece of DNA, and just simply put them together. Or take the DNA, take the fluorescent dye or sensor molecule, and just put it on it. And it requires two moieties, an azide and an alkyne, and also a catalyst. And it's called click chemistry because it works like this, hop, and then it works very fast, if it's done right. And so our pilot study is to develop a method which uses these clicked reads as an alternative to enzymatic synthesis of sequenced probe, yeah. So what we do, we have the target, and this is all RNA. So we are not working with DNA sequencing here, it's just RNA. And we use a solid support because it's very neat to wash off all unbound probes. Then we have synthesized this clickable reads, which are some specific for this particular RNA. And then by base pairing, by genetic code tool, this probe binds to the RNA target. And this is just one step we went. We have a mixture of probes. We added to RNA on the solid support and just wait, like for a couple of minutes. And based on base pairing, these probes will find these unique regions on the RNA target. Then we wash off everything that has been non-bound and add click chemistry reagent. And because we have these groups for click chemistry on each prime end of the probe, we will get a ligated product afterward. Then we detach this product from the solid support and analyze it. And we have all the techniques developed here, how to analyze the DNA and RNA sequences. So this is quite easy. In this pilot study, we designed 50 MER capture probes. So this is 50 nucleotide here to catch the correct RNA from the sample. And then we designed in initial study 6 10 MER probes. So 6 of those. And also some wrong 10 MERs to check the specificity. Some that contain just one single alteration somewhere. And we also added them here to this reaction. And just to illustrate once again, so what happens here is that imagine that you have RNA target which has no mutation, and the second one which has this SNP, or how to call it, mutation, yes. Then we add the probes, and we have 100 of them, but only this that have the base pairing here that are correct ones, they will bind. And afterwards we add the click chemistry reagent, and it creates this long synthetic reed. If there is an SNP, this probe is not going to bind. And this will result in a shorter reed. And then we can, by analysis method, compare, was there an SNP or not? Very simple. It's very simple, but actually hard to make though. It took some time to get it to work, this reaction. Especially with RNA, it was quite a... In order to motivate ourselves, we wanted to select a target which is very relevant for the society and very important one, and hard to detect by next generation sequencing. We considered viruses right away because they are those which are known to use RNA a lot for their survival. So RNA viruses are known to be very hard to detect, hard to treat, but they are mutating a lot. Some examples of RNA viruses are influenza, or not that funny one, HIV, but also the one which has just had this very big outbreak recently in 2014, it's called Ebola. This is an RNA virus which is hard to diagnose because it's very often masking under the influenza. It has very high similarity in genome with influenza. And all the hospitals, clinical wards, diagnostic labs know that it's very hard to distinguish, is the patient having an influenza or Ebola? And the reason for that is high number of mutations that it has, plus similarity to influenza genome. It's quite scary virus actually. It has affected many people, and the survival chance of having Ebola is not high. So it's around 50%. And methods for diagnostics of it are not very good right now. So we created some Ebola targets, amplicon sequence, and also we prepared some RNA. We actually have an actual sample of Ebola RNA, but we didn't bring it here because. So don't worry, we don't have Ebola sample here, but it's in state waiting. Yeah. And next step of this project will be actual work with Ebola sample, trying to detect it by our method and compare it to standard sequencing. This
was before Trump, now Trump is
yeah.
shipping in Denmark.
Yeah, no, maybe, but yeah, it's we are lucky, it's very heavily regulated, so we are not allowed to take it from class 2 lab.
Wait a couple of months.
Yeah, so Ebola target. And our goal was to detect it very specifically if we now have in hand this method and compare it to the existing technique. So what we did, we got the sequence, we synthesized RNA. We also got the actual sample, which is still not here. Anyway, and we created this adjusted probes, as we call them, synthesized them, and then we ran the assay. So here I want to show you the result of this assay. This is a gel, the standard way of analysis for the nucleic acid products. We also characterized this by PCR, by next generation sequencing, which we are also using. Don't get me wrong, it's not that it's not working, it does, but we just want to improve it for the detection of mutations. And also HPLC and mass spec analysis. These bands you can see on the gel, these are these reads, these clicked products. So when we have the correct target, this Ebola RNA target, it gives this big band here, which is approximately 180 nucleotide long. When we have a wrong target, for example, influenza RNA, there is no this band here, so there are some shorter products. And this is what we wanted to see basically, if we can distinguish between the RNA target and the wrong one. This is just to sum up how we ended up actually sequencing 1,500 nucleotides by this method because we overlapped these reads. So imagine again, this is your target RNA, and we have one read, 200 nucleotide, then 200 nucleotide, 200, 200, 200, 200. You can go pretty long this way, given that one can synthesize all these probes, which I'm also going to talk about later on. It's a pain to get that many oligonucleotide probes by existing methods. And when we overlap this simply by the design, then we can easily place them and assign where in this target each particular read is positioned. As I already showed, when there is a mutation, this is our goal, this long read is not going to be created. So we can easily see it by gel that there has been a mutation. And what is good about this method? It is direct. It is directly working on RNA. It doesn't require to get the DNA first, which is nice. It saves time, saves reagents. And we also put fluorescent dyes. We like fluorescence here working. And we could detect it by microscopy as well and quantify our reads. So using microscopy, we could get down to as little as 1 picomolar, I think it's like 5 or 1 picomolar RNA target. 1 picomolar, it's very good sensitivity for viral RNA. And workflow compared to next generation sequence, it took only 2 hours to do that, versus 4-7 days for the standard sequencing. And I put here in a parenthesis, never, because not everything can be actually sequenced by next generation sequencing. Some targets are just not working. And I think that this method could be a good solution for targets like that. So when we have to optimize this assay further and develop a better work analysis. Again, I said this is a pilot study. It's just proof of principle, basically. Read length is crucial. So the length, how many of those clicked oligonucleotides are put together is very important for sequencing application. Human genome has 3 billion of nucleotides alone, so this is a lot. And yeah, I don't know, we have never thought about sequencing a genome by this method, like full human genome. It's now designed for small parts of it. But anyway, bioinformatics says that shorter reads are better, actually, than the long ones. And that makes it simple for chemistry, because it gives better yields to align, to click together shorter DNAs read. There at Stanford, where I collaborate with on this project, they actually now try to do 6-mer, only 6 nucleotide long reads, and then they click them together. The problem with that short reads is that the assay has to be below 5 degrees Celsius in order to bind them to the target. This requires cold room. So this is my collaborator at Stanford who is now trying this out. And all of that, I have personally tried to work in the lab, which is 0 degrees Celsius. I really like, don't hurry up to set up this assay here. It's quite an experience to sit and freeze there. Yeah, anyway, but this is something we are trying now. We are trying to shorten actually each particular oligonucleotide to only 6. Yeah, and then use a library of them, lots of them, to sequence longer.
Sorry, can I
Yes.
ask a question? But I bias your sample in the sample by actually going to lower temperatures? You can also have phase transitions, you can have all kind of effects coming from temperature.
This is an in vitro technique. This
But
is
that
not
means-
going to be, yeah, this is for in vitro.
Which means that not necessarily applies to living, if you have the Ebola in a real being. so-
Serum.
Yeah.
Serum, yeah.
Yeah.
So if you take serum from a patient, it's going to be fine. This guy, he's doing oncology studies, so he's not going to do any in vivo stuff.
But
It's-
it will be different in vivo and in vitro.
It will be nice to compare. Right now, we even cannot compare, right? So if we could just get an option.
So it's very premature.
Yes. It's just something under the development right now.
Maybe, since you're on the slide,
Mhm.
I don't know how the overlapping is done, but the 6-man itself is not particularly specific.
No,
So, but maybe you overlap yourself out of that problem.
yeah, so there are ways to handle that. They're all bioinformatics. So you have libraries, and then there are like barcodes. So it's actually longer. It's 6 murdered bytes, and then it has a barcode. And then by cycles of annealing the annealing detection, you can actually figure out. It's very engineering like work. So a lot of this is not my expertise. So I'm just getting in touch with collaborators, get amazed what they can do there. Like technical, it's microfluidic engineering, some advanced bioinformatics based, how they use this like cycles of annealing detachment. Yeah, but exciting what is going to happen now. And it's all LNA modified, so it kind of helps to specificity. Yeah, we'll see. Yeah, so I kind of skipped this story, so that we have done some other methods, and for detection of mutations, I just wanted to show where the sensitivity is right now. This is the old fluorescence detection of probes, which has been labeled, and we could never get down lower than, so you can see 500 nanomolar concentration. It's very high, it's 0.5 micromolar. When we move the assay to the solid phase and to microscopy, we actually get down to attomolar concentrations of DNA. That you can see the spots here on the slide. These are DNA molecules that are labeled by the dye. Attomolar is actually way too low. Very few species have that little amount of target. So it's way below the limit that is needed actually. So it's quite good to apply microscopy for the detection. And perspective is creating extended library of short clickable probes, optimizing oligonucleotide synthesis for preparation of these sequencing probes. The way we are synthesizing them right now is very high. It requires lots of materials. It's very expensive to do it right now the way we are doing. So we are thinking how to do it in a bit different approach so we can have thousands of these probes easily and cheaper. And then applying microscopy for detection and quantification of reads. So we are working right now on how we precisely quantify. And this is actually an amazing point I forgot to say. So sequencing, when it affects by amplification, it's not quantitative. No one can say how much of the target has been initially. We are trying to quantify it directly, which is interesting. I'm actually curious to know how many of those are in the sample A. And testing more targets will be, yeah, a good next step. Some questions I want to leave you, and I'm speculating about, and please write me if you have ideas on that. So what is the future of personalized genomics, right? So what is it all going to when we have access to all the sequencing data, right? I presented viruses, but it's potentially all of it can be applied for human genome. And how can ultra-fast sequencing methods affect research community, clinical work, and the society? So right now, in order to do this sequencing, you need to send a sample somewhere or go to the doctor and wait for weeks. Imagine if it's there, right, in a minute, right, all the sequences. What will happen? How can we apply that? Is it good? Is it bad? Yeah. I'm definitely speculating about all of that, and want also you to leave you out with this thought. What isn't necessarily that good to have access to all of that data. I want to finish by acknowledgement that this is my group here in Odense. Some of them are actually present. Yeah, I am very happy about working with you guys. Thank you for all the great work. I have not done anything of what I presented myself, so it's all done by their hands. And I'm also very happy about my collaborators and continuous inspiration they are providing. Also, I want to thank NAC, Nucleic Acid Center and University of Southern Denmark. And also the money, this is quite costly, what we are doing. So it's Willem Foundation, National Institutes of Health USA that are sponsoring our work. At the end, I want to say thank you all for this opportunity and thank you for kindest attention. Thank you.
Thank you, Kira, for this interesting presentation that is now open for comments or questions. So please go ahead.
Yeah. I don't know whether this is a little bit too technical, but if I understand right, the core technology is that you sort of, by click reaction, link the fragments of the individual probes, but with the background of the LEU template, the DNA chemistry, we know that these sort of intramolecular click reactions will be very fast and very efficient, even if there are 1 or 2 or 3 mismatches at the sort of dangling ends where you do the click reaction. So they will happily go together. If you have very short probes, like a 6-mer, and you have both sides 3 dangling ends which will perfectly happy sort of do the click reaction, how will you sort of maintain a specificity? If you go to longer probes, you will have more specificity, but still mismatches in the sort of either ends of the oligo will be sort of probably not affect the speed of the click reaction very much, or sometimes even improve because it's sort of more flexible. How is that gonna sort of fundamentally affect the perspective?
Yeah, so thank you. That's a good question. So for long ones, for long reads, we have tested that. So we have this mismatch. Actually here you can see one control. This is line number 6 with the mismatch actually at the end. It's not forming it. But there is a technical step which is called warm wash. So we have warm buffer. And this is LNA that does the trick. So you can increase a bit of temperature of washing of unbound stuff. And makes it very specific. It makes a big difference for longer probes.
So the chemistry would actually work, but you remove the oligo before you actually go into the chemistry.
Yes, and this is a good one. So we had the control, and if there is no LNA there, this is not working. But the big difference for match versus mismatch of like 10 degrees that LNA is giving gives us the chance to wash off on specific stuff. When it's 6-mer softening, what we are looking into is polynomials, G, for example. So this approach which I showed 6-mers, it's not for decoding this mixed-mer sequences. This is not going to be specific. But it's for some certain regions, and it's working very fine. And for example, G quadruple sense, quantifying length. For some more specific sequences, we expect that it can work.
Yeah.
So going back to in vivo, where is the limit, or what cannot be done with clickable elements, or what could be done with clickable elements? Then you said like short reads, you need to have low temperatures, but like for longer reads, the way I understood it, it might be possible. And so basically, just in terms of understanding how clickable elements could resolve really real-time reads of RNA.
So what it can do, it can definitely sequence some short part of RNA. Let's say microRNA, something very, very well characterized. We know where it is, we know what we look for. In this version, it cannot decode sequence without knowing what you're looking into. So you need original actual sequencing data in order to create the probes. If there is you see an effect, for example, some effect of the gene, you don't know the sequence of it or don't have any idea what it could be, then this method is not going to work.
Yeah, but you could detect actual mutation rates at the source.
Yes. Yes.
Okay.
Given that you have an initial genome
Yeah.
sequenced by something else. It's an interesting. We are not on a hurry for in vivo work. 90% of all it's all diagnostic in vitro, so we stick a bit to. But research-wise, of course, in vivo stuff is interesting.
So I didn't quite get the transition from mutation analysis to sequencing full genome. So this is a mutation analysis,
Yes.
is that right?
Yes.
So so this is you isolate the virus RNA,
Mhm.
and then you hybridize your probes, and then you look for gene fragment.
Yes.
Why did you use those long fragments? I didn't get that. Why didn't you use a 10-mer plus a 10-mer?
I said it probably not very clearly in the beginning. So all the detection of mutation is potentially sequencing of the bigger. You can extend it. So here we looked for a mutation, but then got curious how far we can go with it, and we ended up with 1,500 nucleotides. And then we compared it to the technology right now, which is this NGS. It reads 300 at the time, 300 only, and we already did 1,500. So in principle, our point here is that it works very well for mutations, actually, for short regions. But it can't do more than that, probably. It can be used for larger parts.
So for the short probes, where is a mutation best placed? If you have a 6-mer, I agree fully, you have to go for short probes. If you have any 6-mer or an 8-mer, is the mismatch, where does it mostly decrease the click reaction? Towards the end or towards the middle? Also for the short ones?
Yes, absolutely. And
Because you might get all or none hybridization with the very short ones. But Francesco?
I have a more general question. I mean, how predictable mutations are, and they're probably due to complex system or the system is in, but how much is known about the dynamics of mutations per se? I mean, now I understand you want to study once something has happened, and mutations happen, you can like that, but
it's a lot of them happening all the time.
I see.
I want to say number that above 10% of genomes are mutated.
So they're mostly random, or they due to environment? I mean,
So this is the way we adapt, actually. Yeah.
okay. So it's due to the environment, basically.
Yes.
Okay.
This is also why Ebola has a lot of them. It adapts very fast. Yeah.
How many are repaired?
Those which do not benefit, they get repaired. Those that create some benefit and phenotype, they stay because it survives better. This is the general, I guess, Darwinian.
Okay.
Yeah, but first they are repaired and then some stay, and those that stay, they might end up showing Darwinian
Yeah.
advantage.
Yeah.
And then, so that's, I think it's a different kind of repair, isn't it?
There are these neutrals that are happening and then disappear. And some they stay because there's a pressure from, like, for example, we start treating the virus with some drug, and then it develops a mutation that helps it to survive. Those which do not have this mutation will be eliminated, and the one which has will produce more of it, and it will stay. This is how the theory is now.
One more question from Stefan.
How's the actual detection done? I mean, you show a gel now. Gel is not particularly convenient for routine analytics. So how is it actually done? On a gel, of course, you can see whether you have the full length fragment or anything shorter. How is that sort of
The two
envisioned?
fluorescence microscope. So yeah,
But then you would see any length as long as there's a fluorophore on it.
we label it in a certain way that only the full one gets the time. Yeah. HPLC was good, but it's not that sensitive.
No.
But HPLC is easy one.
Okay, thank you very much for showing us your work.